Hello all, welcome to Life Science Library YouTube channel. In this video we are going to see about signaling pathway of ethylene. Under the unit 6 of Iicsirnet Life Science portion, that is plant physiology. This topic becomes very important for Iicsirnet. So many questions came from ethylene signaling pathway since it is very unique. First of all, for any signaling pathway to be clear, we should know what are the components of signaling system. We should know what is the ligand and what is a receptor. Ligand here is ethylene. Receptors are membrane bound receptor. Receptor are dimeric in nature. They have multi-pass transmembrane domain and it also contains copper. ETR1 is ethylene receptor 1, ethylene receptor 2, ethylene response sensor 1. These receptors are present in endoplasmic reticulum. It contains copper. These are previous year questions. Membrane bound receptor previous year question. Copper presence previous year question. Hope kindly make a point of it about this previous year questions. These are the components of signaling. Next, about the presence of domain. Let us draw ER membrane. Consider it as an ER membrane. Let us draw the membrane bound receptor. Histidine kinase domain. Receiver domain. They have disulfide bridges and they also have copper. These areas are histidine. It's not histamine, it is histidine. Histidine kinase domain. And this area is receiver domain. This is how receptor looks like. This is histidine. Histidine is an amino acid, histidine kinase domain. It is homologous to bacterial two-component system. Bacterial two-component system. This is the exact picture of signaling receptor in ethylene. So it is a transmembrane domain. We have an ER, it is present in the ER lumen. This is cytosol. This is endoplasmic reticulum membrane. It is a membrane bound receptor. This is overview about receptor. CTR is nothing but constitutive triple response 1. It acts like serine threonine kinase. These are the basic details about the receptors. This is the two domain of the receptor, histidine kinase domain and receiver domain. Next we are going to see about the entire flow chart of the signaling pathways. The whole story is based on the presence or absence of ethylene. The entire pathway acts differently in the presence of ethylene, in the absence of ethylene. First of all, before going into pathway, one basic thing we should understand is ethylene receptors. Ethylene receptors are negative regulator. This is also a previous year question. Easily we can get 4 marks in part C. Ethylene receptors are negative regulator. Please make a mark of it. This is very important. Ethylene receptors are negative regulator. Now we will go into the pathway. First we will see what will happen if ethylene is absent. What will happen if ethylene is absent? First we will draw ETR. Ethylene response, triple response 1. It is receptor, ethylene receptor. In the absence, it is active. In the absence of ethylene, ethylene receptor is active. Then it activates CTR, constitutive triple response 1. This also activated in the absence of ethylene. Then comes E1, EIN2. EIN2. This is active. This is active. This leads to phosphorylation. This leads to phosphorylation and inhibit proteolytic cleavage. Inhibit proteolytic cleavage and fragment migration into nucleus, which leads to polyubiquitinylation. Polyubiquitination and proteasomal degradation of EIN3. Ethylene response gene turned off. No transcription occurs. EI13 degraded in nucleus. This is one side story. Hope this is clear. I will repeat it again. In the absence, what will happen in the absence of ethylene? ETR1, ethylene triple response receptor. It becomes very active. So it is called as negative regulator. It is active in the absence of ethylene. Then it activates CTR, constitutive triple response 1. It also becomes active. Then comes EI1, 2. It becomes phosphorylated. Inhibit proteolytic cleavage. They inhibit proteolytic cleavage and fragment migration and leads to polyubiquitination. Then EI13 enter into nucleus. It is degraded. No transcription occurs. This is unique pathway where a component enter into nucleus and it gets degraded because of the absence of ethylene. Even in the absence of ethylene, it is a unique pathway that receptor becomes active. CTR1 becomes active. So it is very unique. So it is called as negative regulator. So ethylene is called as negative regulator plant hormone. This is one side story. What will happen in the absence of ethylene? Now we will see what will happen in the presence of ethylene. Quite exact opposite. ETR1 becomes inactive. ETR1 becomes inactive. CTR1 has also become inactive. CTR1 also becomes inactive. EI12. No phosphorylation occurs. No phosphorylation. So no phosphorylation and no phosphorylation occurs. Because of no phosphorylation, cleavage takes place. Cleavage occurs. And C-terminal domain will migrate into nucleus. Will migrate into nucleus. No, this polyubiquitinylation, proteasomal degradation will not occur. Transcription occurs. This is the major difference. In the absence of phosphorylation, cleavage occurs. Because of this cleavage, one end will enter into nucleus, that is C-terminal end, and transcription occurs accordingly. And series of reaction occurs, where EI12 is converted to EI13, EI13 then to ERF1, then ERG, then series of reaction will occur. This occur in nucleus. This becomes unique. In the absence, receptor become inactive. This is unique. We will sum up now. In the absence of ethylene, ETR1, CTR1 active. They are active. EI12 is phosphorylated and inhibit cleavage. It undergo, then EI13 in nucleus undergo degradation, that is polyubiquitinylation mediated proteasomal degradation. In the presence, ETR1, CTR1 inactive. EI12 is not phosphorylated. Cleavage occurs. C-terminal domain will enter into nucleus. EI12 will go into convert into EIN3, then to ERF1, then to ERG, then series of reaction occurs. This is about the signaling pathway of ethylene. Point to be noted down is, in absence, they are active. In presence, they are inactive. In absence, we have phosphorylation that inhibit cleavage. In presence, no phosphorylation of EI12. Cleavage occurs. This is the whole idea about signaling pathway of ethylene. This variation terms to call it as negative regulator. Hope this video is very clear. About CT, interesting fact about CTR1. These are constitutive mutations. In case of mutations, these are constitutive mutations. On constitutive mutation. So what is constitutive mutation? They produce response even in absence or presence. Constitutive mutants. Constitutive mutant produce response in absence or presence. It is seen in CTR1, ETO1, ETO2. This is an important previous year question. What is the constitutive mutant's response? It gives response even in presence or absence. Its constitutive mutants is sometimes seen in CTR1, ETO1 and ETO2. It is an important previous year question. Then what is ethylene? Insensitive mutant. It is another question. What is ethylene insensitive mutants? It is ETR1, ETR2, ETN1. Sorry, EI. Ethylene insensitive mutant. Ethylene insensitive mutants. ETR1, EIN2, EIN3. These are ethylene insensitive mutants. This is also an previous year questions. First, now we will see about few abbreviations that we have seen in this pathway. That is very important. What is ETR1? What is CTR1? ETR1 is ethylene receptor 1. CTR1 is constitutive triple response. This is about the elaborations of ETR1 and CTR1. Hope as an additional content we have seen about constitutive mutants and insensitive mutants and abbreviated form of all these. Then as a part of signaling system, we have seen what will happen in the presence of ethylene, what will happen in the presence of the absence of ethylene, and we have seen why ethylene receptors are called as negative regulators. It is not ethylene is a negative regulator. Receptor of ethylene are negative regulator. Ethylene is a positive regulator. Ethylene receptors are negative regulator. This is previous year question. Please make a note of it. Hope this video is clear. Thank you for watching this video.